First of all, thank you very much for inviting me to present here. I found it a pretty cool mix of different biologists, mathematicians, physicists, and so on to be in the same pot and kind of have different view and solve biological questions using their expertise. So this is pretty cool, and I hope you keep on doing this in the future. So we heard a lot about how DNA is packaged into the nucleus and also transcription and DNA repair. And now I think it's about time we have a look at DNA replication and how DNA replication is involved into genome organization and genome architecture. And I will give you a showcase study of what we found for the role of DNA replication in this process. So first, before I start, I would give you a very brief biological overview. So we are all on the same page. I work with the B cells, and these are our antibody-producing cells, and they do this by generating mutations in the antibody encoding genes, so the IGH locus here in the example, and they do this by expressing a DNA mutator enzyme which targets the antibody encoding genes, generating mutations and breaks. And this is very important. Audio is fine, because sounds weird. And this is very important to generate the vast repertoire of antibodies in a process of antibody maturation, which allows us to fight the different pathogens we are exposed to. But the downside of this process is that they can get downregulated, misregulated, and can target also other genes in the genome, generating mutations, and this can lead to double-strand breaks and translocations. And we have very often cases where the antibody encoding genes are translocating with proto-oncogenes like MYC in this example. And MYC-IGH is a hallmark translocation of Burkitt lymphoma, and this is a very aggressive type of lymphoma. And as you know, lymphoma is one of the most common types of cancer. And the Burkitt's lymphoma is prevalent in some areas of Africa, so it's a not very nice disease, yeah? So we want to, of course, find therapies for this, understand the mechanism, find therapies, and also prevention strategies. And in our lab, we were interested in understanding the mechanism of this process. And what we did a screen in B cells using shRNA, so knockdown screen, to identify factors which modulate the frequency of IGH-MYC translocations. And we identified the MCM complex to be important for the translocation. So when we deplete the MCM complex, we have a reduction of the translocation. So what is MCM doing? As you know, we heard already, this is the replicative helicase, which binds to origins of replication and unwinds the DNA to initiate replication. So we deplete MCM up to 70–80% in our system, and in this situation the cells are normally proliferating and we don't detect any major changes in gene expression, but we have a phenotype, so what is the defect? So we thought that by depleting the MCM complex we change the initiation landscape, so which origins are initiated upon S phase. And to test this, I used an assay called short nonsense transsequencing, where I where I isolate the newly synthesized DNA strands and map them to the genome, and this would give us the location of the firing origins or initiation of replication. And when we deplete MCM, we have a very striking picture. So here I'm showing you tracks of short nonsense sequencing, and we are overlaying the wild type and the MCM knockdown situation. And what we saw is that the normally active origins in a wild type situation become decreased in activity, and then we have activation of origins in normally not so active regions of the genome. And this is not actually a random thing, and we could overlap it with different chromatin marks, so we can see that the normally active origins in a wild type situation, they are in an actively transcribed open chromatin, as you can appreciate by the pro-6 transcription marks, the active chromatin marks, and then the not so active origins in the heterochromatin become activated upon depletion of MCM. So you can see that we have increase of origin activity in the heterochromatin regions, which are the big compartments. And this is a very interesting result, and we follow this in a different story, which if I have time, I would tell you a bit more about. But in the case for the translocation, what this means, so MYC is actively transcribed region as the IGH locus, and what we saw is that there are origins of replication in MYC, so upstream of the MYC promoter, and when we deplete MCM, we lose the origin activity, so it's decreased, and this correlates with less binding of MCM, and this we observed via chip. And in this situation, we see a decrease of the IGH-MYC translocation, and also when we check translocation globally using nanopore sequencing, we saw that when we deplete the MCM, we have a significant reduction of the MYC translocations. But this is only a correlation, so we wanted to see if there is a direct relationship between the origin activity and translocation frequency. And to do this, we decided to delete the origin of replication upstream of MYC. And we observed that when we delete the origin of replication, we also have this significant reduction of the MYC IGH translocations and also the MYC translocations genome-wide. And when we reintroduce the origin of replication downstream of MYC, so this is 14 kb downstream of MYC, we could restore the MYC IGH translocations and also the MYC translocations genome-wide. So this showed us that the origin activity is directly involved somehow in the translocation genesis. So what is the mechanism? How does origin activity play a role in the translocation genesis? So we know that the origins fire throughout S phase. We have some origins which fire in early S phase, then in mid S phase, and then in late S phase. So what this means is that some regions of the genome get replicated earlier, and then we have some regions in the genome replicated late in S phase. So we thought that by deleting the origin or decreasing the origin activity, we would modulate the time when MYC is being replicated, and this would have some implications later on with genome architecture. And to check this, we performed an assay called replication timing sequencing, and we observed that normally MYC is early replicating gene, and this is the situation where we get the translocations. When we deplete MCM, we have an asynchronous replication. So MYC become asynchronously replicated, and this is where we have the decrease of translocations. And when we delete the origin, we saw a very interesting phenotype. So we saw that MYC becomes late replicating. So we generate a domain where MYC becomes late replicating. And in this situation, we have the translocation defect we saw. And when we reintroduce the origin, then the timing is restored. MYC becomes again early replicating. So indeed, when we delete this origin of replication, we change the time when MYC is replicated. And then this leads to defect in the translocation frequency. So we have replication timing somehow playing a role in the genesis of the translocations. So what is the role here? And we know that replication timing, so the time replication timing is associated, so we don't know too much how, but it's associated with genome architecture. So we have early replicating regions, which are in the euchromatin, late replicating regions in the heterochromatin, and they also have this, their specific localization in the cell nucleus. So we wanted to see if the changes in replication timing would lead to changes in the genome organization. And when people do replication timing experiments and they see defects in replication timing, they usually do high-seq to check the genome architecture because of this very nice correlation between early-late timing and AB compartments. And this is the first thing we did to see if there is some changes which would somehow explain the replication phenotype and translocation defect we saw. And I'm not going to explain the matrices because we saw them often enough now, but we did high-C in all our conditions. So here we have the wild type, MCM depletion, and I'm showing you the MYC locus. So this is a 20 kb resolution. And as you can see on the first side, we don't see any dramatic changes in the genome organization. So the TATs are conserved, everything is fine, compartments are there, and we have the very striking phenotype in the replication timing. So this is the track here, but this is not corresponding or correlating with any changes in the AB compartments, and this is the zoom in here. And the same is true when we delete the origin. So we have our late domain here, but we don't see any changes in the AB compartment. So here is again zoom in, and also the TAT architecture is conserved. And the same is true when we restore. So when we reintroduce the origin and we restore the timing, we don't see any changes in the genome architecture. So this shows us that okay, changing the timing there doesn't show any differences in the local architecture in mix. So what causes the defect? So HiC is a good method to see interactions within the chromosome, but it doesn't have this high resolution to see interchromosomal contacts. Yeah, I was mixing up these interchromosomal contacts. And in this case, I have to tell you, we have around about 1 billion reads. We sequence this, so it's a reasonably good depth. And the contacts between MYC and IGH, so the translocation we are interested, are only 3 reads. So HiC is really not the assay to use to check whether by changing the replication timing, we change the frequency between MYC and IGH translocation. So we needed an assay where we can target, in a targeted way, see if replication timing would change somehow the organization and the frequency. And how we imagine this, so here is some fish picture. So we probe MYC and IGH. And if MYC and IGH are in close proximity, we have the translocations. And if they're not in close proximity, we don't have a translocation. So we used an assay called 3C, where we use probes for the IGH and MYC to see the contact frequency. And what we saw is upon MCM depletion. So when we reduce the origin activity, we have a decrease of the contacts between MYC and IGH. And this is also true when we delete the origin of replication, decrease of contact frequency, and then when we reintroduce the origin of replication, we restore the MYC and IGH contact frequency to a wild type level. And this led us to our model, which is that basically MYC and IGH they are normally in neighboring chromosomes, and they are both replicating early in S phase. So what we think is happening is that in a normal situation, upon initiation of replication, they initiate replication within the same replication or DNA repair hubs. So when you have double strand breaks and then they're in a close proximity, this will facilitate the translocation genesis. And when we modulate the time when MYC is being replicated by depleting the MCM levels or by deleting the origin, then MYC becomes replicated at different time points, so it would not share the same replication repair hub, and that's why we decrease the frequency of translocations. And this is all very cool, but it's one translocation in one cell type, so we wanted to see if the mechanism is global. And to do this, we actually used acute ILL, so the acute lymphoblastic leukemia. This is also a very severe type of a cancer, and it's predominant in the kids, so pediatric cancer, and it's one of the most common leukemia cancers. And why we selected this, so what happens here is that we have a fusion with the MLL protein, so it's a fusion protein which would drive the oncogenesis. And why we selected this is because MLL and its fusion partners are both replicating early. So if our model is true that you need the same synchronized timing for a translocation to occur, we need to find genes which replicate at the same time. So we used this model system to see if replication timing is also true there. It's also important for this process. And we depleted MCM in the leukemia cancer model by using SH-MCM knockdown. And indeed, what we saw is that normally both genes are early replicating. When we deplete MCM, one of them becomes late replicating, so we desynchronize the timing. And in this case, yeah, it's a bit tough to read assay. In this case, we get a significant reduction of this translocation frequency. This is a digital droplet PCR assay where we score for the translocation events. And to summarize basically what I told you, DNA replication timing is a global mechanism. So what we see, we don't only see this in this model, but we checked a few different leukemia and lymphoma models, and we see the same when genes which are translocating replicate at the same time, and we modulate the replication timing, then we decrease the translocation frequency. So meaning that the replication timing is a global mechanism in this process. And to summarize what I told you, so I used the MYC-IGH translocation to study the mechanism of oncogenic translocations, and we found that the MCM complex is important for this process. And what does MCM do? So there we have origins of replication within the translocation partners, which modulate the time when the genes are replicated, and by depleting, so decreasing origin activity or deleting origins, we modulate the time when the genes are replicated, and this influences the translocation frequency. And also this led us to hypothesize that replication timing is important for the translocation genesis by bringing genes into close proximity. And yeah, I think I was very fast, so I have a few minutes to tell you a bit about the second story. So we became, after this story, we became interested how replication timing is regulated in B cells, because we saw this very nice phenotype when we deplete MCM. So I can give you a bit more detail now. So we have the B cell, and we use this assay called replication timing sequencing to check when the genome is replicated. And normally a wild type situation is the blue one. We have regions replicating early and regions replicating late. So the chromosomes are replicated in temporal order. And when we deplete MCM, we lose this kind of temporal segregation. The cells become asynchronously replicated. And this was very interesting for us because there is another global replication timing regulator, and this is RIF1. And what we know about RIF is that it binds to heterochromatin and suppresses the activation of origins there. And when we knock out RIF, we have a very similar phenotype to what we saw upon MCM depletion. So we were wondering how MCM levels and RIF regulation interplay in the B cells to regulate the replication timing. And very long story short, we had very surprising findings here. So first of all, we saw that RIF doesn't bind to the heterochromatin in B cells, but it binds to the active chromatin, and it's actually important for the initiation of replication. And this was at first very surprising result and very contradictory to what is in the literature, but then we partnered with David Gilbert and Sarah Bonomo, so the RIF specialist, which generated the RIF knockout data, and they also confirmed in their studies that basically in the B cells the phenotype is completely different, and indeed RIF has this very novel role, so it binds to active chromatin and is important for the initiation of origins there. And what we kind of model is that in order for the actively transcribed chromatin to be early replicated, you need combinational binding of RIF molecules and MCM loading, and this would aid the higher probability of and earlier replication in the actively transcribed chromatin. And in the silent chromatin, we have less MCM molecules binding and no RIF binding, and this kind of confers lower probability of firing of these regions, and that's why they replicate later in S phase. And if we split the genome into bins based on their replication timing, so we have the early replicating regions, which represent these regions where we have RIF binding and MCM loading, and then the late regions would be the regions with no RIF binding, and we speculate a less loading of MCM molecules. And the interesting phenotype, what we saw is that when we knock out RIF, yeah, I'm sorry, so I didn't fix this model. So imagine that there is no RIF here. So when we knock out RIF and when we deplete the MCM molecules in these early regions, there is no RIF binding to activate the origin firing, and we have less loading of MCM molecules, so this actively transcribed open chromatin becomes later replicating. And what we see is also that in the silent chromatin, we have a compensatory advancing of the replication timing. And this is how the data looks like, so we have kind of a complete flip of the replication timing, so the normally early replicating regions become later replicating, and we see this compensatory advancing of the replication timing. And this is a story which is on bioRxiv, and I'm looking forward to any advices here, and if you have any ideas about how to explain the mechanism, I would be very happy to talk about this. And what we just want to conclude here is that in the B cells we have two mechanisms which would confer the early replication timing, and this is the loading of MCM molecules and the activity of RIF. And we think this is very important to safeguard kind of the early replicate, to safeguard and mediate the early replication of the actively transcribed genes, because when we have a delay of replication timing, this would be associated with accumulation of mutations and some cancer transformation. So that's why we think the cell develops kind of this dual mechanism to protect early replicating regions. And with this, I would come to my acknowledgement slide. So this was done in the lab of Roshat Pavri, who is here in the back. And for this story are important 4 people, but most vote to bioinformaticians. So this was a very sequencing heavy paper with a lot of novel analysis we needed to do. So these were the main heroes in this story. We collaborated with a lot of different groups throughout the world for the RIF story, David Gilbert and Sarah Bonomono. And this is IMP, where I'm doing my research, and it's a cool place to do science, a lot of interesting people, so please visit, and thank you.
That was very interesting. It was very, very enjoyable. I have a couple questions that I'm curious about it, these early replications. On the first part, why do I need MIC? If you remove MIC and then you just have to put MGM alone. Afterwards, you're able to reactivate without MIC, right? That's what you say. You remove.
Let's go and see what you mean. Here?
No, first, you go, when you do the experiments, you first remove.
Yeah, you have to tell me.
First, you remove MCM.
Yep.
And then you lose the early replication.
Yes.
Then you remove MIC. Nothing happens. You still but then you only add MCM. You don't have to add MIC again, right? And you still go back to have early replication, right?
You mean basically
when you restore, you
yeah.
just restore.
So the only thing we are modulating is the origin activity. So in one case, you have an origin which is upstream of the MIC promoter, so we don't touch MIC at all. We don't change the expression, nothing, so we checked all of this.
So MIC is not involved with the replication,
exactly.
right? So
yeah.
it's just the origin is there, right?
Yeah, exactly.
That's what I'm trying
to...
Yeah,
exactly.
Okay, that's what I was trying to figure out.
Yeah.
On the second part of your talk, you said you have these 2 mechanisms because you want to make sure that the B cells sort of don't stall because one of the mechanisms is not working. But there's some negatives. Why don't I have 3 mechanisms or 4 mechanisms? I want to be safe. So what's the bad part that comes by creating alternative mechanisms?
Yeah, I cannot answer to this. Yeah, I don't know if there, of course, probably there are some other regulators, right? But what we saw and was striking is that when you...
They're completely independent, right?
Yeah, exactly. I would say that basically the regulation, we were struck because the phenotype we saw of MCM depletion and RIF knockout was very different to what you see when you deplete MCM and what you see when you have a RIF knockout. So this was kind of a complementary mechanism. So kind of both of them act in some way together to give this flip of replication timing. That's why we were thinking it comes from different kind of regulative pathways, but it could be, I mean, what we know is that RIF can actually activate, or it's important for the loading of ORC molecules in the G1 phase, so it has kind of this promotive role. To have more loading of ORCs and MCM molecules, and in the S phase it has this kind of suppressive role, so we think that maybe when we knock out RIF, then we have less stable ORC MCM molecules, so you start already with a kind of depleted system, and then when you load less MCM molecules, then you increase the phenotype, make it more severe. So this is kind of our working model, but no clue.
Okay.
Very nice talk. Two very nice stories. So there are hot spots of translocations.
Yeah.
How we can is there any, connections with the timing of replications, or how we can explain that?
You mean what we do is we know what are the kind of this AAD translocation partners. So AAD is the enzyme which generates breaks and mutations in the genome, and it's responsible for the translocations. So what we see is AAD off-target genes, and we can basically correlate them with the replication timing. And this was really very interesting because what we saw is that all AAD off-targets are early replicating, and basically when we deplete MCM, we have this decrease of origin. So we think that in this case would be really a global kind of defect. Not only IgH-MYC in the B cells, but all other AAD off-targets would give you the same. Or maybe I don't get your question.
Okay, thank you.
I think that this work was excellent. My opinion is that this breakthrough in an understanding of the origin of chromosome aberration, tumor-associated chromosome abnormalities. I have only one question. Can you say the methods for determination of translocation 814, so-called c-MYC IgH rearrangements? The methods, only the methods.
To detect the translocations.
Yes.
Okay.
Yes.
So
you have said that you have found increased translocation
yeah.
after this moving.
So what you can do in this kind of system, we are very lucky because, as I told you, let's go to the general slide. AAD generates the mutations and their error-prone process to get the breaks. So we know where AAD is targeting because we can do this mutation analysis. And we can design probes which are outside of the AAD targeting region, and in such a way that when you design a probe here upstream of the breakpoint, upstream of the IgH point, and when you get the PCR product, this is a translocation. So it's a very specific thing. We do this kind of a nested PCR to detect the translocations, and also what we do is for the genome-wide sequencing to detect translocations, and this is this one here, too fast. What we do is we generate breaks within MYC, and then we allow for a translocation to occur, and I enrich for the MYC translocation and do the nanopore sequencing. So it's basically enriching for the translocated region and sequencing. Yeah.
I was wondering whether you considered another possibility, which is related to the fact that when you deplete MCMs to that extent, you actually lose the dormant origins. So it was shown by Julian Blow and by Juan Mendez that under these conditions, if you have a replication stress, for instance, you will see that the cells are unable to activate these backup origins.
Exactly, yeah.
So could it be that when you don't have dormant origins and you have, in a very tiny subset of cells, you have a replication fork posing at the MYC locus, for instance, the fact that you don't replicate it from a dormant origins would actually prevent the translocation? You know, it would be a totally different mechanism, but It's a bit counterintuitive, but formally it should...
Of course, we don't rule this out, but what we saw was really the proximity. So whether it comes from the replication timing or this passive replication, we cannot say.
Because the replication timing phenotype, you see it globally at the population level, but you don't know where those cells that undergo the translocation.
Yeah, exactly. This is true for the case of MCM knockdown. There we have this global asynchronization. But in the case of the deletion of the origin, we introduce local change of replication timing.
I agree.
So, yeah, I mean, it could be really passive replication. I cannot say. We haven't tested this, but yeah.
Okay, thank you. Last question, quick answer, please.
Hi, nice talk. I don't know whether you have, probably you mentioned, the number of breaks that you get at the MIC region is the same all over the type of experiment you did or not? Do you know anything about that? Because my question is that if it is the same, or if it is not the same, because I believe that those translocations occur between the MIC and the S regions, right? Where you have G4, R loops and all that, and this is influencing. Then I was wondering whether when you have early replication, you might have more R loops, more G4, so that you will get more breaks. And when you have late replication, transcription is already gone, and you will not have that. Therefore, that might
this is what we were wondering, whether it's R-loop related, so by removing the origin you would have less kind of this R-loop formation. But what we do is we generate Cas9 breaks in the MIC locus. We generate Cas9 breaks. And then this is downstream of the double-strand break formation. So we have already double-strand breaks, and we do this genome-wide assay to check for translocations. And by making double-strand breaks with Cas9, and this is with high frequency, we are not able to restore the translocation. So it's really downstream of the double-strand break formation. And that's why we thought it's the synapsis or localization. Yeah.
Okay, thanks a lot. So we should all thank Michaela.